You are welcome to my YouTube channel. Topic of discussion is virulence factors of pathogenic bacteria. Pathogenic bacteria are disease-causing microorganisms that require virulence factors to cause diseases. Virulence is a degree of pathogenicity, and pathogenicity simply means the ability of the microorganism to cause disease. What are these virulence factors? The virulence factors are both structural and non-structural factors which aid or help pathogenic organisms to adhere, invade, multiply, survive in host tissues in order to establish infection and subsequently diseases. Some of the structural virulence factors include capsules, membrane, that is the form or they are part of the morphology of the pathogenic organism. The non-structural virulence factors include enzymes and toxins. Adherence of pathogenic bacteria to host tissues. This is simply the attachment of these pathogenic microorganisms to host tissues or cells. In order to cause infection, you first of all have to attach to the host cells. Some of the structural virulence factors include pili and fimbriae. They are simply hair-like structures which are found on the surface of the bacterial cell, whether they are motile or non-motile. So the pili, this pili aid attachment. For example, Neisseria gonorrhoeae has pili on its surface, cell surface, and it uses this to attach to the epithelial cells of the genital tract. Then you have the capsule, is another structural virulence factor. It's a slimy, viscous material which surrounds bacteria. This is a slimy, viscous material that surrounds bacteria. For example, Streptococcus pneumoniae, there are two strains. We have the capsulated Streptococcus pneumoniae strains, which are known to be pathogenic. And the pathogenicity is attributed to the presence of the capsule, while the uncapsulated or non-capsulated ones are non-pathogenic. Invasion of host tissues. Pathogenic bacteria make use of special proteins known as invasins in order to forcefully gain entry into epithelial cells through a process of endocytosis. This has been seen in studies done with Salmonella species. The non-structural virulence factors include enzymes and toxins which break down host tissues. The first one is phospholipase. Phospholipase is an enzyme that is known to hydrolyze phospholipids. Phospholipids are found in the animal cell membrane, and if they are hydrolyzed, this leads to destabilization of the cell membrane. The cell membrane is charged with the responsibility of maintaining the integrity of the cell. It is a semi-permeable membrane and selects what enters the cell and what goes out of the cell. If the cell membrane is destabilized, then cell death occurs. Coagulase is an important non-structural virulence factor used by Staphylococcus aureus. In fact, it is responsible for its pathogenicity. Coagulase is known to convert coagulase is known to convert soluble fibrinogen to insoluble fibrin, which this pathogen, Staphylococcus aureus, use to cover itself. That is, the fibrin clots form are used to cover cells of pathogenic Staphylococcus aureus. This is because it's trying to avoid being phagocytosed by macrophages and neutrophils. Next, you have the lecithinase or alpha toxin. This degrades lecithin into phosphorylcholine and diglyceride. Hemolysin and streptolysin O are known to destroy red blood cells. Other enzymes include hyaluronidase. Hyaluronidase is known to degrade hyaluronic acid. This hyaluronic acid is the interstitial cement that is found between cells in tissues. So it is known as a spreading factor. Once hyaluronic acid is degraded by hyaluronidase, the pathogen is able to spread to other parts of the body. Then you have the kinases. You have examples, streptokinase, which dissolves fibrin clot. And this has been found useful in medicine. It helps to dissolve clots that may be formed in cases of thrombosis and also in stroke patients. And you have IgA proteases. IgA proteases are known to bind to IgA at the hinge region. The IgA, specifically secretory IgA, is known to protect the mucosal surfaces of the respiratory tract, the gastrointestinal tract. For example, Neisseria meningitidis is known to produce IgA proteases, which inactivates IgA and is able to establish itself and cause meningitis, bacterial meningitis. They have collagenase. Collagenase is known to break down collagen, which is present in muscles. Collagenase is produced by histotoxic Clostridia, which are known to cause gas gangrene. So these are anaerobic bacteria, and these include Clostridium perfringens, Clostridium novi, Clostridium septum. Then you have neuraminidase, which breaks down neuramic acid, which is found between epithelial cells in intestinal mucosa. So once neuramic acid is broken down, the organism is able to spread to other parts of the gastrointestinal tract. We have other virulence factors such as protein A, produced by Staph aureus is able to bind to IgG. Protein M is produced by Streptococcus pyogenes. Lipopolysaccharide is found to be involved in the pathogenesis of gram-negative bacteremia, causing fever and also other symptoms. Then you have the toxic shock syndrome toxin 1, TSST1, which is produced by Staph aureus, that's been involved in causing toxic shock syndrome. Then you have the Pathogenic exotoxin A, which is produced by beta hemolytic streptococci. Other virulence factors are exotoxins, which are associated with diarrheal diseases. For example, in cholera, which is caused by Vibrio cholerae, it's known to produce an exotoxin. And then staphylococcal food poisoning, an exotoxin is produced, which is actually an enterotoxin. Thank you for listening.